update automatically. And you will notice that I don't have, the only organisms I have selected are vivax and falciparum. And so you can always go to your organism preferences and modify this further. So let's say I want to add null-z to my results. I can go ahead and select one more organism, add it to my results, and then go back. There's this little feature here, this little arrow back to your site search. If you came from a site search, you can go back to it, or you can use your browser return arrow. But I'll go ahead and just go back here. And so admit a couple of people back into the room here. Okay. And so now you'll notice, hopefully if everything worked out, that we also have Plasmodium null-z here. Okay. The third thing that we explored was the gene page. And the gene page, again, hopefully, I'm just going to pick a gene. So let me go here, and let's select one of these genes, this hypothetical one up here. So we're looking at this gene that's hypothetical. So in theory, there's a gene there, but its function is hypothetical. But there's a lot of information on the gene page that may help you decide for yourself what this gene may be doing and where it may be working and functioning. And so again, if you haven't explored this, I would encourage you to go to your favorite gene page and explore what's on that gene page. There's a lot of information. For example, we can go to the phenotype section right here, and you'll see that there's a CRISPR phenotype experiment. This is in particular in Toxoplasma gondii. And this is an experiment where every single gene was knocked out using CRISPR, and then they determined the fitness of each of the resultant knockouts. And you can see here, this is the fitness graph of all the genes in Toxoplasma gondii GT1 in this case. And your gene of interest, this blue one, it happened to be because the one I selected from the previous window. It's one of the most fitness conferring genes in this particular genome. So you can see that it's extremely important, if you want to take that as essentiality, it's an extremely important gene for the survival of this parasite at the stage where this experiment was done. So now, even though this is a hypothetical gene, I know it's essential, right? So this is very useful information. So now we know that it's an essential gene. And there are other features in here as well. I can look at its cellular localization, for example. So I can, or where is that? Actually, this is for another strain. So I'd have to convert it to another strain. So it wouldn't be on this page here, which is fine. I can look at the protein properties and features and explore if there's any information here. Some cases there isn't, right? So sometimes it's hypothetical for a reason because there wasn't a lot of information available for that gene, which is okay. We can look at the mass spec evidence. So you can see that this gene was found in secretomes, for example. It was also potentially phosphorylated, so we can take a look at that under post-translational modification. And you can see that this gene has multiple phosphorylation sites. So now we know it's an essential gene. It looks to be a highly phosphorylated gene, right? Which is another interesting thing that we found about this gene. We can look at its expression based on transcriptomic data and see if it has anything interesting. So anyway, not to belabor the gene page again, we've already gone through it, but just to point out that there's a lot of information on a gene page, and if data is available for a particular gene, it'll be displayed there, and it's a way for you to then explore this data on a gene-by-gene basis. One thing that I wanted to make sure you're aware of, that many of the items that show up on the gene page are also searchable genome-wide. So don't feel like you can only do your searches gene by gene. So this is something that we tell people in all of our workshops. If you ever find yourself doing something over and over again, sort of the same thing over and over again in the database, send us an email. There might be a way to do it automatically in bulk, which would really save you a lot of time, and I would encourage you to do that. So for example, this phenotype data, I can go under searches and search for phenotype. I was demoing this with my group. I can go to CRISPR phenotype, and now I can say, well, I want anything that is between -5 and -6.89, which is the most fitness conferring. And so you run the search, and now it returns 368 genes that are really down at the bottom end of that graph that I showed you before. And so now, instead of doing it gene by gene, you can do it in one shot, and you have the fitness score for each of the genes that were knocked out. So the last thing that we did today was the search strategies, which we're doing right now, right? These are single step search strategies, but I could have, for example, now these were done in a strain called GT1 in Toxoplasma. If you work on Toxoplasma, you would know that there's another strain called ME49, which a lot of the experiments are, it was the first genome that was sequenced. So a lot of the experiments that were conducted in Toxoplasma were mapped to ME49. So maybe you'll decide, well, I want to know if these 368 genes have orthologs in ME49, so we can use this transformation trick and look for orthologs. I'm going to select ME49 and select ME49 right here. And now this will return orthologs of my 368 GT1 genes that are essential. And now these are my ME49 genes. And so by orthology, you may say, well, if they were essential in GT1, they're probably essential in ME49, in the other strain. Right. And so now you can build this strategy and ask, well, how many of these are potentially secreted, for example? And this is just an example of types of searches you can run. So I'll just go ahead and run this just to quickly demonstrate this. But very quickly, you can build yourself a in silico experiment, tethering different searches together and combining them together using intersects, unions, and various different tools. So as you can see here, there are 13 genes that have a signal peptide, so potentially secreted, and were essential in GT1. And then you may look at those, and these may be some interesting effector proteins. And as I indicated, you're the scientist, so you want to make sure you look in here and see what results you get. I'm glad to see a dense granule protein, which is good, but then you see other things potentially in there that don't make sense. And so just like in a gel, you'll have positive controls and negative, and you may have some background showing up in your gel and so forth. You should treat these results similarly, basically. One thing I wanted to highlight, and I'm just going to jump to maybe another. Let's see what I have here. So what I wanted to highlight here, I'm going to go ahead and focus on this strategy. So you remember that we talked about the intersect and the union operations, but there are also some, minus operations. So you can subtract this result from that result or vice versa. And so something to think about. So that's very functional. We did this co-location. Hopefully you appreciated that the co-location is pretty much like if you knew the GPS coordinates of all the houses in your city and all the cars in your city, you can now co-locate them and say, well, give me any house that has a car within 500 meters of the front door, right? Because you know the GPS coordinates. Same thing in a genome. The genome is your world, right? And the location on the genome is your GPS coordinate. And so anything that can map to the genome with a coordinate, a genomic GPS coordinate, basically, you can co-locate. So you can co-locate a gene because we know its coordinates, and you co-locate a SNP, which is, we know it's a single nucleotide polymorphism. So now, if you know their locations, you can say, well, give me all genes that have a SNP in the 500 nucleotides upstream of the start site, right? So that was the utility of this co-location. But also notice that we have these options, which we didn't talk about, which are these ignore step, the previous step or the following step, which is quite nice. So instead of having to rerun the strategy without, let's say I want to ignore this step from my strategy, I can quickly click on here and say, ignore step 2 and revise. And so what's going to happen now, it's going to show me the results, what the results would look like if I take this transmembrane domain step out of my strategy, basically for the temporarily. And then you can go back and say, oh, interesting. I wonder what happens if I ignore step 1. So now you know how many transmembrane domain proteins. In this case, I think I searched for things that have orthologs in mammals or something in humans, maybe humans. So that's a nice way for you to ignore one or the other step. Okay, so this is, in a nutshell, that's what we did today. Hopefully you were able to appreciate that. I would encourage you to go back to the exercises. I know some of you probably did not get a chance to do everything in the exercise, but that's okay. These exercises could be quite dense. So I would encourage you to try them out on your own and come back to us with questions. Tomorrow, what we're going to be doing is we'll start at 8:30. The Zoom will open, but we won't actually start the sessions until 9 a.m. And we're going to spend pretty much tomorrow on the genome browser and interacting with the data in a browsable manner. And specifically, we will look at all types of data sets or data types that you can load map to the genome, and then use that information to help you guide you in terms of knowing if a gene structure is correct. Many of us end up cloning genes or asking your graduate students or postdocs to clone a gene, and you want to make sure that they're actually not cloning a gene model that, even though it's in the annotation, that gene model may be incorrect. And we are at a stage in the development of these of the data that's available in these organisms, is that we can actually look at the real data. We can look at the RNA sequence data. We can look at transcription start site data. We can look at chip or chip-seq data, and we can make our own conclusions about these genes. And we can say, well, you know what? I don't trust the annotation. That official annotation is really not good. There's a missing exon here based on the data. Or the start site probably is not here. It's probably, the gene may be much bigger or much smaller based on the data. And so then you can start making your own conclusions about the actual gene models and be a bit more clever in your strategies downstream as you do your laboratory experiments. Okay, so this is all I was going to do for the wrap up. Any other comments from the other instructors or any questions from attendees? Anything specific you want to bring up right now?
Maybe, Omar, just to note that that if people encountered problems or difficulties, things that didn't make sense, we have several more days to be able to review those, and in breaks and in various times we should be able to do so, or of course staying on after the call today or at some other time. If you still encounter a problem that is that you don't understand, or something that is unresolvable or seems to you to be wrong, click on that Contact Us link at the top of every page in the database, and send a screen picture or describe in terms or text terms or both what it is that you're encountering, so somebody can get back to you and make sure that you can use these resources to maximum effect.
So unless there are any other questions, obviously people feel free. You're welcome to leave the Zoom, and we will see you tomorrow bright and early or late at night, depending on where you are in the world. And of course, if you do want to ask questions right now, you know, feel free to hang on for a few minutes, and we'll be happy to answer any questions you may have. All right. See you tomorrow, everybody, if you're leaving.
I have one question, if I may.
Yes, of course.
If you have a list of genes for a specific organism, and I would like to check if there is data in the database concerning to the RNA expression and protein expression from the same genes, is that possible to see?
Yes, absolutely. So let me, I think I'm still sharing my screen. Yes, I am. So take you back. Let's pick vector base to change views a little bit. The best way to do this, if you have a list of genes, is you can run a gene search for gene IDs, or you can just type ID right here and it'll show you where the search is. And now what you can do is you can paste your list of IDs in here in this box. And let me actually go back here and let me get some IDs for you just so we can demo it. And I'm going to pick anopheles. I'm just looking for genes that have a single peptide, so just to get a list of genes. And I'm going to download. So this is giving you a nice demo of the download feature. If you just want the gene IDs, I can clear everything here, include one transcript. And I want this. I'm just going to show it up in my browser. And so I'm just going to copy these here. And let's go back to our searches. And I'm going to look for ID. And now back here, let's say this is your list of genes. You can copy and paste it right here. And then you can click on Get Answer. Okay. So now what's going to happen is it's going to return a step in your strategy that includes, so apparently I copied 67 genes. So I have my 67 genes of interest. And now I can add a step here. Well, so there are many things you can do, different ways you can interact with this. So one is I can add columns, right? So I can, let me close these old ones that I had open. But I can, for example, go to my columns, and if I knew of a specific experiment that I want to load in Anopheles, so this was Anopheles, so you have to specifically select the Anopheles experiment. Let's see if I can find one quickly. Here's an Anopheles experiment, and I'm going to load the the graph. So this is loading both the sense and the antisense transcript, and I'm also going to load the percentile graph, and then I'll click on update. And so now, since this is Anopheles pest, once these update, I can quickly now see for a particular experiment what my genes are doing, and I can see the expression patterns. You can also load the values associated with these genes for each of the stages, so it really depends on what you want to do. The other thing you can do is I can add a step and say, well, I'm going to add a step and search for genes based on, let's say, RNA-seq, and let's pick RNA-seq. And obviously, this is, vector-based has a large number of genomes, and sometimes you'll see that there are lots of data sets here. And so one thing you can do is you can use the organism filter that I showed before. You can set it just to Anopheles, or right here, I can just start looking for the strain of Anopheles pest, and then this will show up. Now it filters my data to Anopheles. And so now I can pick the experiment that I'm interested in knowing what my genes are doing. And then you can be very specific. So for example, this is a blood meal experiment. So I can do a differential expression, for example. This had biological replicates. And I can say, well, I want to know if any of my 67 genes are differentially regulated by at least twofold. And you can change the p-value, let's say to 0.05. So I want to know if any of my genes are regulated by at least twofold when I compare the 3-hour after blood meal to no blood meal fed, and I can run the step. So now what will happen is it tells me out of my 67 genes, 5 of them are differentially regulated by twofold in that particular experiment, right? So now you kind of, in a bulk way, you get a quick answer for this. And obviously you can modify this step as well. We also, depending on the database, we have proteomics data. And so I know we're just starting to load this data in vector-based, but it's increasing quickly. And so now you can see here that there's peptides mapped to genes from various experiments. Of course, it's not as comprehensive as RNA sequence data. So we can go ahead and add, let's say, this brain proteomics experiment. And I can ask, do any of these genes have at least one peptide from this brain proteomics experiment? And the answer is 0 here, but it's not surprising because I only had 5 genes here. But then you can also, as I showed you with our steps in the strategy, I can actually ignore step number 2 and ask, well, out of these 67, how many of them have peptides? The answer may still be 0, but at least this one, it wasn't 0, it was 12. So 12 of my genes also have peptides from this brain experiment. Okay, does it make sense, Gabriel?
Perfectly. Thank you
Cool.
very much.
Sure. Do we have any other questions, anybody? So yeah, if there aren't any more questions, obviously feel free to sign off. And I will hang on here for another 15 minutes. Other instructors also, if you're tired and you need to go and stretch or something, feel free to leave. I'm happy to hang on today, but up to you. So.
I'll have another question if I may.
Of
Sorry.
course, yeah. No, absolutely.
It's a great opportunity.
Yeah.
So it is possible for us to filter all by expression levels in terms of RNA. I mean, if I have a value like FPKM or TKM, it's a variable there. So I want to filter to say this group of genes have a higher FPKM, or I mean readout of expression level than this group of genes. Or if I put the same list of genes, I can do a sort by the level. Is that possible to do?
It is. Yeah, yeah. No, absolutely. So we do have, let me see if I can demo this here. So again, with any search, so yeah, there are a couple of ways of doing that. So one thing you could do is, and let me change this back to an intersect here. So with these 5 genes that are also, in this case, we had expressed the intersection, you know, I can, and this experiment is this is the experiment. I'm just gonna copy, maybe I think I'll copy this name here. I think then I can find it easily in the table. So I can add a column here, and I can look for, that didn't work. Let's see, oh, here we go. So this is the experiment, and you will notice that these are the normalized, we have the normalized values basically available for each of the samples in this experiment. So if I load all of these, I update. So now for at least for these 5 genes, because this is an intersect here, it will load additional columns here with the values. And these are all sortable. So depending on what you're interested in, again, depends on the organism you're on and the experiment, but you can then take a look at them. This was an experiment, looked like with biological replicates. So each sample is listed here separately. But then you can sort, you know, based on, you know, higher to lower or lower to higher. So that's one way of doing it. The other thing you can do is in your search. So you could, for example, start off with a search in, let's go ahead with RNA-seq. And let's go to test, for example. And with any of these experiments, you'll notice, and we'll cover this on Tuesday, I believe. We're going to be doing some on Wednesday, we'll be doing some transcriptomic and proteomic searches. And we will be exploring these different types of searches. But you'll notice, depending on the data and what was available, you'll have different types of searches available for each data set. Most of them will get a fold change and a percentile search. Some of them will get a differential expression search if they have biological replicates. And then yet other ones, if they're strand specific, you can also do sense anti-sense searches, like find me all genes that are going down in expression in this sample, while in the other sample, the antisense expression is going up, right? So if you're trying to find genes that are potentially regulated by antisense expression. So if I pick a fold change experiment right here, you can see now that I can select based on the samples that I select. Let me pick one actually that has more. Let's see if I can find one that has more samples in it. I don't know what this one is. Here we go. So we'd have to look at the details of this experiment, obviously, but there are many samples in here with these names. So we have to look at the data set itself to find out, but let's pretend we know what these are. So these look like controls, control 1, 2, 3. And so what you can do in this search here is you can select samples from this experiment to compare to each other. So let's say I'm going to pick these VBN samples, and I'm going to compare them to, you know, let's imagine that this is the right thing to do. I'm going to compare them to these 5 samples right here. And you'll notice that this graph here gives you a cue as to what kind of gene would be returned. So in this case, I'm asking for a protein coding gene that's up or down regulated, so differentially regulated by at least 2-fold. When I compare the average of these samples, the blue ones, so you'll notice this box is blue, so these blue samples to the red samples right here. So that's what I'm asking for. So the 2-fold is between the average of these to the average of those. You can play around with this, and you can obviously make this higher. You can say, well, I want at least 4-fold, right? And so now this is giving you a cue as to what this looks like. I can change this to, instead of up or down regulated, let's say I just want to find genes that are upregulated when I compare these to each other. And now I can also switch this back to average, for example. And so now I'm saying, well, I want to find genes that are upregulated in my red samples, so these comparison samples, compared to my blue samples, these reference samples. And I want this upregulation to be between the average of the samples, and it has to be at least 4-fold upregulation. Or you can even make it more stringent and say, well, you want to compare the sample that had the maximum expression value from the blue samples, compare it to the sample with the minimum expression value, and you want that fold change to be 4-fold upregulation, right? So this would be the most stringent one that you would identify. And so once you figure out the parameter that works for you, and that's giving you the right answer. So there were 636 genes that met this criteria from the samples I selected. Obviously, I didn't look at the details of the experiment, so these could be completely bogus. But let's say these were important. Then what you can do, and we can see here that this is matching. I picked samples that were on this side of the experiment versus that, and so you can see that in general, I'm getting the right profile, which is good. Then what you can do is you can add your genes of interest, right? And see if your genes of interest intersect with any of these. Did they meet the profile that you had selected before? Okay.
Yeah,
hopefully
yeah,
that helps.
great.
Yeah.
That's very, very helpful. Thank you.
Good.
Can you go, once you're already there, can you go into Genome View and explain for us a bit about in that same experiment? So
Okay. Yeah. So this one here, this Genome View.
Yes, exactly.
So what this does, it's going to take all the genes from my search. So there's 636 genes right here, and it's going to paint them on the chromosomes, or if there aren't chromosomes for your organism, it'll be contigs or scaffolds, whatever is the largest entity from that organism. In this case, we're looking in anopheles, so you'll see that there are the chromosomes and the different right and left arms of the chromosomes, and it'll paint the genes on that chromosome. Blue, the genes are on the strand going from 5' to 3' left to right, and then red are the reverse strand genes. And so then you can very quickly. This is not statistical, obviously. You're not trying to look for statistical, clustering of genes, but simply gives you a quick and dirty way for you to see where these genes are located. And so depending on what you're asking, you may then, for example, if you're looking in something like Plasmodium, where there's variable genes, or in trypanosomes, for example, you would, and your search ended up returning genes that are actually VAR genes or VSC genes, you might find that they're clustering in certain parts of the chromosome, as would be expected, like in subtelomeric regions and stuff like that. And so that's a nice and quick and dirty way for you to see the distribution of the genes across the chromosomes.
Yeah, I have used this to do exactly like this, but what I have interest to do is to check if I have some cluster of genes inside the same chromosomal unit. I work with Leishmania, by the way.
Okay.
So is that possible?
Yeah, and Leishmania you can do it as well, but again, just as a reminder that this will not be, because there are methods out there for doing statistical clustering, right? Because just because genes are close to each other on a particular chromosome, it doesn't mean that that's significant, right? So and that really depends on right.
But I'm just interested in location.
Yeah. So absolutely. So let me. Oh, I must have clicked something here. Capture. Let me. Not sure what I'm activating on my browser. Let's try this again here. And let's do a new window, and let's go to TripDB. And I'm just going to run a search for Leishmania genes that are, again, let's just try single peptide. That's an easy one to do. And which Leishmania are you interested in, or it doesn't matter?
Phantom.
Doesn't matter? Okay.
Infantum.
Oh, Infantum, sorry. So let's pick Infantum. There we go. So these are the genes that have predicted signal peptide. Great. And now if I click on Genome View, you will see the same thing. So you see the distribution, but Also note in your results, we do have the genomic location provided here as well, right? So this is coming straight from the what's called the GFF file, or at least that's where this comes from, this data. So it gives you the indication of the location of the gene and also its orientation, whether it's on the plus or the minus strand. So there are a couple of ways where you can get that. And you'll notice here that some of these are like a wide bar, and that means that the genes are close enough to each other. There are many genes in this region here. And so if I mouse over that, you'll see that, I don't know if you can see my mouse over, there's some text that show up and it tells you that this region has 6 genes in it. So to me, this would be more of a clustering effect, than any of the other genes. And then if you click on it, this will give you some information about these genes. So there are 6 genes here, and then you could take a look at them. I thought it used to give you the product. Oh, maybe if I mouse over, let's see. Yeah, so this is a hypothetical protein. They're probably all going to be hypothetical.
That's Leishmania.
Yeah, exactly. They look like they're all hypothetical, but they're probably, I wouldn't be surprised if they're maybe it's a gene duplication region, quite possible. Here's a surface. The first one is a surface protein, a GP46. So it's quite possible that those are all maybe also surface proteins that are duplicated.
For instance, if I want to know what are the genes between these 2 genes, is that possible? I mean, you take the gene location and you can tell me, I want to know what genes are between gene X and gene Y.
Right. So for that, I don't know if we have a way, and the other instructors on the call may remember or not, I don't think we have a way to do it easily, at least with the search strategies. The way you could do it is by looking at the gene itself in the genome browser, which we will look at tomorrow anyway. But let me, oh, I clicked on the wrong thing. Let me go to one of the genes. Sorry, Suzanne, I think you were going to say something as well.
I did say JBrowse, which you've already said.
JBrowse. Okay, perfect. So I'm going to click here and go to, let me see if I can go to one of these easily. Or let me go back to the results. It's easier to click here. So I'm going to just click on, let's say you're interested in knowing, you have these 2 genes here and you're trying to find out what's in between them. So what you can do is you can take the coordinates of these genes. So you want to go from, in this case, 110,000 something. So I can copy this, and you want to go all the way to 131,000, let's say. So what we can do is we can go to the gene page. I'm just going to go straight to the genome browser from the gene page. And so this is going to load a default view with one of the genes, that first gene in view. So must be this one here in the middle. And now what I can do is I can modify the numbers here. So I want to go from 110,000. And let's take this down here. And I think it was like 100 and I don't know, let's do 35,000. I'm just being now. Yeah. And so now when you click on go, it's going to load everything in that region. Okay. And so then you could see all the genes that are in that region. So you can obviously. Can I have it as a list of gene IDs? So can you have it a list? Oh, so no, you can't, but there is, now that you ask it this way, I thought of something that you can do on the website. So if we go back to the homepage, so I can look for genes based on genomic location. Here we go. So let's try location. So I can select genomic location. And so now I can go and select Leishmania and Phantom. Where are you?
Go down.
There we go. There we go. And now, and then I can select, in this case, I think we were on chromosome 3, it doesn't matter, but I'm going to select, let's put 110,000 to 100 and let's say 40,000, get answer. And so now this is going to give me all the genes that are in this region. So there were 7 genes in this region. And so here's a list of genes, and you can download them and so forth. Yeah.
Thank you. Thank you very much. That's very
sure. Good. Yeah. Good. I forgot about that tool. That's after a while it comes up like, oh yeah, you can do that.
That will be easy. I'm just interested to see some what have between some genes that I have characterized here in our lab.
Okay, great. All right, I think we're at 1:30, and it's possible that other people on don't have any questions. Everybody good? Okay, then thanks, Gabriel, for your questions. Those were very, very nice, and hopefully you can make it tomorrow, and we'll see you tomorrow. Same to everybody else.
Yeah,
so
I have a lot with the Go function.
Great. Excellent. Okay, goodbye, everybody, and see you tomorrow. Bye-bye.